So today, I'm going to be talking about quality checks and maximizing success of natural enemies. And before we get started, we'll just do a quick run through of some upcoming events. So for the TunnelTalk series, we have a roundtable coming up on July 24th and August 28th. We'll have a panel discussion on greenhouse structures August 14th. And then September 4th, I just confirmed that we'll be doing a winter spinach production session. So keep your eyes peeled on social media for the registration information on that. We're continuing along with the Understanding Alternative Nutrient Amendments series through OFCAF. The schedule got a little bit jostled because we had some technical difficulties last week. So every session just kind of got bumped down 1 week. So we're still going to talk about all of the same things with all of the same presenters, but just kind of 1 week later than that original post. Nova Scotia Young Farmers is hosting their summer event July 27th. And then Acorn is hosting a gathering at Abundant Acres on July 28th so that we can all be jam-packed of in-person stuff, but it should be good. Okay, so today I'm going to be talking about biological pest management, and we're going to be focusing a little bit more on the insect side of things compared to the disease things. I had a bit of a hard time assigning a title for the session, specifying that it was about insect management and not necessarily disease. So I hope no one is overly surprised or disappointed by that distinction now that I've kind of been able to focus that topic a little bit better. I'm definitely not an entomologist, so we're really just going to be reviewing some of the resources that are available to us about maximizing the success of our biological control agent, or BCAs. And so for people who are more interested in learning about specific control options, certain predators or parasites to tackle their certain pests, definitely recommend looking out to your insectary or reaching out to me on a separate space. This is going to be a bit more general as opposed to tackling specific problems. So we'll start off super easy. Why are we sort of seeing biocontrol become more prevalent in the industry? There's a few reasons why. There's definitely more interest and knowledge to support an ecosystems approach to producing our food and agriculture. So as opposed to just growing one crop one way, we're thinking more about, the soil life that goes into that as well. We would like to see insects in there, but just ideally not the wrong ones. So yeah, we're kind of thinking about it more collectively as a whole, as opposed to just sort of an isolated system. It can also result in a reduced time and labor spent with regular spraying. So if you have a spray program, you could be in there once a week. But in theory, if you're able to do some sort of slow release of a BCA, then you shouldn't have to be in there doing the sprays in addition to the scouts and the crop work that has to happen as well. It also reduces the amount of residue on vegetables, fruit, and flowers, which is always a good thing. And we are setting ourselves up to be a little bit less reliant on conventional chemistries. Integrative pest management doesn't say that we cannot use conventional products, but really it's just about using all of the tools in our arsenal. So looking at cultural control, making sure that we are being very mindful about varieties we're selecting and, where we're putting our cull piles and that sort of thing, and using all of those tools to also reduce the pressure in the first place. So by the mid of the season or end season, if we do need to come in with a conventional spray, that's okay. We've done a lot of work to make sure that we only have to use it once. The big sort of mindset behind that is really trying to slow down any sort of resistance development that we might see. So that's happening in both disease and insect populations. And so this is a pretty common schematic that I've got here that I'm sure you've probably seen somewhere before, but we've got our one red resistant individual in that first generation before pesticide application. We apply our pesticide, and now we've actually got 30% of the population is resistant as opposed to a much smaller number. And then as time goes on, as those individuals kind of reproduce and further along generations, you'll notice that we have more of these resistant individuals as we apply another spray. We're looking at about 80% of those individuals now being resistant. So by not using these conventional chemistries, we're sort of able to slow down that process, which is a good thing. So there are three general groups that biocontrol agents basically fall under when it comes to enemies of insect pests. We have predators, and so these are actually feeding on our pest. They're less host specific, so typically you can release one and expect some level of control over a handful of our insect pests, and they consume many pests throughout their life cycle. The other group is parasites, and so in this case, the insects are depositing eggs in or on the pest, and they're typically only eating one single pest in their life cycle, and that's usually happening as that egg sort of hatches or the early life cycle stages sort of emerge from that host pest. Now, parasites are extremely host specific, as you can imagine, so you have to be very sure of what you have going on in your crop before you want to release a parasite. Otherwise, it's really not going to do anything for you. That last group is pathogens, and so these are going to cause fatal disease of pests. We're not going to talk much or at all about pathogens today. I think that kind of falls under the realm of the biological spray programs that we had talked about earlier in the year. So it'll be pretty much focused on predators and parasites today. And so I did include just a quick screenshot from the OMAFRA sort of general information on use of beneficial organisms. So we have a list of our target pests that we're typically going to see in protected settings and also outdoors. And then they've broken it down by type. So you have a predatory mite versus maybe something parasitic. And then the species that are available to us to help tackle that problem. So this is a great place to start. Once we've started identifying issues in the greenhouse, then at least we can kind of narrow down that list and start to get some better information about what our options are. So in terms of releasing them, in terms of releasing these BCAs, we have a few options. Sachets is a very common one, and it's a rearing system that contains multiple life stages of that BCA. And so in theory, that should facilitate a continuous release of our natural predators under the optimum conditions. Obviously, these aren't going to be able to release indefinitely, but they can offer multiple weeks of protection from just one sachet or unit area. So these sachets typically contain bran. Alongside that, we have a non-pest or a food mite. And so the feed mites or the food source for our predators are going to feed on fungus growing in that bran. They typically come with a hanger, as you can see on some of these photos, or they come with a stake. So they are intended to be either hung in the crop or sort of stuck into a growing container if you have something that's a little bit smaller and not a like indeterminate tomato, for example. And the type of sachet that you're going to need is actually going to depend on your growing conditions. So these are sort of formulated for either low humidity or high humidity environments, whether they can withstand sort of direct or regular moisture or whether they can't. And so that's going to impact the sachet that you receive, and it's going to impact the success that you have in that space. So it's always worth mentioning what growing environment you're looking to control your pest in to know what would be best suited for you. The other option we have is broadcasting. And so in this case, the predators are mixed in with some sort of carrier and then distributed through the crop as is. They can be spread across the canopy via blowers, and there are some handmade options or some more commercially available options depending on what your cup of tea is. But you can also do that manually as well. So you're just sort of shaking at your container as you walk through the crop and trying to get a somewhat even distribution of that product. There are some rumors of distribution of BCAs through drones. I think that's probably pretty early, and we can save that for another day. So on average, broadcasting is typically done every 1 to 2 weeks, just making sure that you're maintaining that population size. You can supplement your BCAs, so you can purchase pollen or other supplemental food that you can apply to the canopy just to make sure that those predators sort of have the juice they need to keep going. But we are still relying on a more regular application compared to the sachets. Research on vegetable crops, such as sweet pepper, it has shown that sachets can provide consistent and higher numbers of these predators compared to broadcasting. And I also think that it's worth mentioning that the sachets are also less labor intensive. If we're going in there on a weekly or biweekly basis, we're really kind of mimicking like a spray program. And ideally, if we don't have to do that, that would be good. I will say though, broadcasting is typically a better option for young crops or those with like a sparse canopy. Certain biocontrol agents that can't fly, which there are a handful of those, it's really difficult for them to travel plant to plant, especially if we don't have direct contact between either the product or the leaves from one plant and the leaves for the other. So there's definitely a place for broadcasting in our biocontrol program, but it's just about figuring out what makes sense for you and what makes sense for the crop that you're growing. And so before we talk too much about proper release of predators and parasitoids, there are a few sort of quality control checks that I think are worth discussing. So successful biocontrol is dependent on a number of factors. Obviously, we want those natural insects or those natural enemies to be good quality and sort of ready to work when we release them out into the crop. Unfortunately, as with any living organism, these products are subject to variability, and that starts right at the insectary, all the way through that shipping, storage, distribution, and emergence process. So one of the first things we want to pay attention to when we're receiving a shipment of these natural predators is the condition of the package that we've received. A big one, especially out here on the East Coast, is has there been a delay in shipping? I know a few years ago when there was an issue with the highway being closed, there were a lot of shipments of these BCAs that were basically thrown out immediately because they had been sitting on those trucks for a few weeks and they were not meant to do so. So definitely pay attention to when you've ordered something and when it's supposed to arrive and when it does arrive. And just sort of keep that in the back of your mind when you're opening that package and considering how successful that particular batch is going to be. Some more obvious things would be looking at the package. Has it been damaged throughout the delivery or transit process? Are all the containers sealed the way that we would want them to be? If we look and see condensation or bugs sort of sticking to the inside of that container, that's an indication that something is just not quite right. Obviously, if we can smell something a little bit yeasty, that's not a great indication that the conditions for transit were what that product needed. And also if we're seeing any clumping of that carrier material as well. And so all of these things, like I said, are indications that the quality of the product that we're receiving may have been compromised since leaving the insectary. And we don't necessarily need to wait for our pest damage to get out of control before realizing there's an issue. So there are some things we can kind of do early on just to kind of have a better idea of how well this may or may not work for us and whether we need to intervene in a different way. And so I grabbed this screenshot from Beneficial Insectary. It's sort of a summary of the tools that they would recommend for someone who wants to assess the quality of their biocontrol agents. A lot of these things are readily available. The microscope is obviously a bit more of a specific piece of equipment, but if you want to get serious about sort of regular release of biocontrols, this is a great way to glean some important information on the quality of the product that you've received. The other piece here we have is an aspirator, I believe, is what they're trying to show on the top here. There are videos or plans, I guess, if you want to make your own aspirator. And so we're going to go through a few videos here, if you bear with me, and we're just going to review some of the work that Vineland and OMAFRA have done on checking the quality of biocontrols here. So we'll start off with parasitic insects. I think I have to share my screen though. Hold on.
We'll demonstrate and discuss best practices for quality checking parasitic wasps. Note that these methods are only meant to provide an approximation of package totals and may not be as accurate as a quantitative assessment done by the supplier in a scientific laboratory. Always keep good records when conducting any quality checks on biologicals. It is a good idea to create a standard record sheet that makes note of the species name, package size or type, date received, and the company batch number. Record the date you initiated quality testing, the method used, the number of samples taken, the number of biocontrol agents counted, and any other observations on the appearance and performance of the product. Parasitic wasps for aphids such as Aphidius and Aphelinus are shipped as loose mummies in vials. When you receive your shipment, place a vial from each batch number into a separate fine mesh screen cage and open the lids. Also place a small piece of cotton wool drenched with sugar water or a few drops of diluted honey in each cage as a food source for emerged adults. Place the cages in a shaded area in the greenhouse and count the emerged adults every day as you aspirate them into a vial. Record the daily counts on your record sheet. Immediately release the adults in the aspirator into the greenhouse near your problem areas. The adults will emerge within a few days of receiving the shipment. Diglyphus isaiah, a parasitic wasp for leaf miner, is shipped as loose adults in a vial. Shipments of Diglyphus isaiah should be quality checked and released as described for Aphidius and Aphelinus. Because Diglyphus are shipped as adults, they will start to emerge as soon as the vial is open and can be counted immediately. Parasitic wasps for whitefly, such as Encarsia formosa and Eretmoceras species, are shipped as pupae glued to cardboard hooks or loose pupae in blister packs. When handling the cards, be careful not to touch the area that the pupae are glued to, as you may dislodge or damage them. When you receive your shipment, select at least 3 cards from the package. Count the number of empty pupae on each card using a microscope, and write this number directly on the card with a marker. Also record this information on your record sheet. Place the cards in the crop, making sure to place the card in a way that the parasitoids can easily emerge. Mark the location of these cards using colored flagging tape so you can easily find them again. Collect the cards after 2 weeks and count the number of empty pupae on each numbered card again. Calculate the number of parasitoids that emerged from the cards by taking the difference between the final count of empty pupae and the initial count when you first began quality control. Multiply the average emergence per card by the number of cards in the package and compare this number to the amount on the package label. You can find a link to a PDF showing the calculations in the video description below. For an alternative method to quality check parasitic wasps on cards or in blister packs, place 3 of the cards into a separate, screened-in container. Add a piece of yellow sticky card into the top of each of the containers. Place the container in a shaded area of the greenhouse for 2 weeks. Then count the number of adults that emerged and are stuck to the sticky cards. An even distribution of adults on the cards suggests good flight capability. Multiply the average emergence per card by the number of cards in the package and compare this number to the amount on the package label. To check your crop for signs of aphid parasitoid activity, find an area with known aphid activity and look for swollen and immobile mummies. Aphid mummies will be black if you're using Aphelinus, or golden if you're using Aphidius species for aphid control. To look for signs of Diglyphus parasitization of leaf miner, look for greenish blue or black pupae in the leaf mine under a microscope. Emerged Diglyphus leave round emergence holes in the leaf, whereas leaf miner leave crescent-shaped holes. To check for signs of Encarsia and Eretmosceras activity in your crop, examine the underside of leaves for parasitized whitefly pupae. Unparasitized whitefly pupae are white, and Eretmosceras parasitized whitefly are yellow or tan in color. Greenhouse whitefly parasitized by Encarsia are black, whereas Bomecia whitefly will appear black and yellow.
All right, so next up, then we are going to look at the quality control of predators.
Demonstrate and discuss best practices for receiving and quality checking predatory insects and mites. Note that these methods are only meant to provide an approximation of package totals. It may not be as accurate as a quantitative assessment done by the supplier in a scientific laboratory. Bulk predatory mite biocontrol products for thrips and spider mite control are shipped in bags, tubes, or buckets in a carrier of peat, vermiculite, or bran. When you receive your shipment of predatory mites in a bulk carrier, roll the container over in your hands to mix the contents, or transfer the product to a larger container for easier mixing. Using a small scoop of known volume, such as a quarter teaspoon, which is 1.2 milliliters, immediately take a sample of the carrier media and arrange the sample in a line under a microscope. Using a small paintbrush, sift through the sample and count the total number of live adults and nymphs of predatory mites in the line. If you are checking a product that contains feeder mites, note the difference between feeder mites and predatory mites. Feeder mites are slow-moving, milky-colored mites with long hairs, while predatory mites are fast-moving and smooth. Be sure not to count food mites during your assessments. Take care to separate the pieces of carrier well using the paintbrush, as mites may hide between pieces. Repeat this step for at least 3 samples taken from random portions of the container, recording the counts on your record sheet. Calculate the average number of predatory mites you found per sample and estimate the total quantity of predatory mites in the package. To determine this, divide the total volume of the package by the volume of the scoop. Then multiply this by the average number of predatory mites in the samples you counted. You can find a link to a PDF showing the calculations in the video description below. Predatory insect biocontrol products are used for a variety of pests and are shipped in an equally large variety of formats, including buckwheat hulls, paper strips, burlap, peat, and wood wool. When you receive your shipment of immature predatory insects, place a container in a fine mesh screen cage and open the top. Inside the cage, place a piece of cotton wool drenched with sugar water or a few drops of diluted honey as food for the emerged adults. Put the mesh cage in a shaded area in the greenhouse. Return to the cage daily to count the emerged adults in the cage as you aspirate them into a vial. Record these counts on your record sheet. Immediately release counted adults into the crop canopy near problem areas. When you receive your shipment of adult predatory insects, or a mix of adults and mobile immatures, place all material from the vial or bottle in a fine mesh screen cage. Inside the cage, count all the adults and immatures leaving the bottle as you aspirate them into a vial. Release counted insects into the greenhouse near problem areas and record the total on your record sheets. Alternatively, you can inspect the contents of the bottle near the problem area. Carefully remove a part of the carrier material from the container onto a white sheet of paper or tray. Count the number of moving insects you find in this sample and allow the adults to disperse into the greenhouse. Repeat the sampling until all of the carrier material is inspected, adding up all of the counts to determine the total number of adults in the container. Record this number on your record sheet. Note that Aureus and Atheta have mobile, wingless, immature stages. Include any of these quick-moving individuals you find in the carrier media into your counts. For larger package sizes, like Delosia coriaria, formerly called Atheta, only count a sample of the package. Transfer the product to a larger container for mixing. Mix the product and immediately take a 30 milliliter sample using a measuring spoon. Spread the sample on a white piece of paper or tray. Count the adults and mobile immatures while collecting them using an aspirator. Repeat this step at least 3 times. Calculate the average number of predators per sample and estimate the total quantity in the package. Determine the total volume of the product. This should be listed on the original container. Divide the total volume of the original package by the volume of your sample. Multiply this by the average number of predators in the samples and record this number on your record sheet. To check for predatory mites in your crop, tap the foliage and flowers over a white pan or notebook, as you would do if you were scouting for thrips. Look for fast-moving mites using a hand lens. Alternatively, you can use something as simple as your smartphone to check for mites. Run your phone along the foliage of the crop, and dislodged, light-colored mites will be visible running on the dark surface of the black screen. To check for signs of predatory insects in the crop, look for larvae, nymphs, or adults on plant foliage in problem areas. Plant taps are always a good way to check for active predators. Slow-moving Feltiella and Ophidiides larvae are found in spider mite and aphid hotspots respectively. When you can find all life stages of a predatory species in or near a problem area, that is a good sign that your biocontrol program is functioning well.
All right, guys, bear with me. We're we're halfway there. So we've talked about predators, we've talked about parasites. Next up, I think it's important that we talk about the quality assurance of slow release sachets. These are a little bit of a different beast, where basically we want to track that slow release and see if those generations are advancing the way that we want them to be. So that's going to look a little bit different compared to what we had just watched there.
This video will explain how to assess the quality and performance of slow-release sachets. Predatory mites are excellent biocontrol agents of several pests. They are formulated as loose material for broadcasting over the crop or as slow-release sachets, also called breeding sachets. In contrast to weekly broadcasts that can easily be adjusted as needed, growers rely on the sachets to keep performing well for an extended period of time. Therefore, it is a good idea to monitor the performance of the sachets. However, there is conflicting information about how a grower can determine if the sachets are okay. If you rip open a few sachets at various time points, you should be able to find live predatory mites and prey mites. Here's what they look like in the bran. But keep in mind that their numbers are not good predictors of the performance of the sachet. What is more important than what is in the sachet is what is coming out of the sachet. These are the mites that will disperse among the crop plants in search of pests. The best measure of performance and quality, therefore, is to monitor the weekly walkout. Sachets are like little predator production factories. The initial population will reproduce for several generations, and excess predators will leave the sachet through a hole. Over the course of several weeks, hundreds of predators will come out of the sachet. If you put these numbers in a graph, it will look like this. Weekly walkout of predators increases to a maximum and decreases after that. There is a natural variability between sachets in weeks, and depending on the climate in the greenhouse, the walkout curve can have different shapes. Here is how to monitor weekly walkout. In order to get a reliable sample of the whole box, use a minimum of 6 sachets. Place the sachets in a small, heavy glass on the bottom of a plastic container. You can put multiple sachets together, but if you put too many, it may be difficult to count later. Fill the container with water so the glass with the sachets is surrounded by water. Add a small drop of soap to break the surface tension of the water so the mites will sink. Place the containers in the greenhouse in locations where sachets would normally be hung, so they are exposed to the same conditions, preferably in the shade. This will give a realistic measure of how the sachets are performing in the crop. Top up water as needed. Count the mites in the water at least once a week. For this, you can observe the mites directly in the water, but it is probably easier to filter the water and count them on the filter. Remove the sachets from the glass and rinse the mites from the glass into the container. Pour the water through a small coffee filter. Take care to swirl the water around to rinse all the mites into the filter. Count the mites under the microscope. If the filter with the mites is not too wet, you can put them in the freezer to count later. This also kills any mites that may still be alive. Make sure to label everything and to take notes. The predatory and food mites are easy to distinguish from each other under a microscope. Predatory mites are egg-shaped without spots, while food mites are more irregular in shape, have long hairs, and depending on the species, have spots.
All right. So at this point, we have done our quality checks. We have a good sense of what is in our sachet or what is in sort of our shipment from the insectary. And now we want to make sure that we are releasing them into the crop in an effective way. So this will be the last video for today, and then we'll jump back to the slideshow.
Practices for receiving a shipment of biocontrol agents and how to release them into the crop. The first step is unpacking the shipment of biologicals. When opening the box, the contents should be cool. There should be no excessive moisture and no smell of ammonia or yeast. If something doesn't seem right, you may want to check the quality of the biocontrol agents before releasing them. You can find more information on this by following the link at the end of the video. Keep bottles and tubes in a horizontal position until use to prevent compression of the contents and to make sure the natural enemies don't suffocate. It is best to release the biocontrol agents immediately. If you have to store the product, check the storage conditions on the label or the supplier website. Note that a normal fridge temperature is often too cold. In this case, you may be better off keeping the product at room temperature, or use a cooler with ice packs. Humidity is very important when it comes to the storage of many biologicals, especially for predatory mite sachets. It is better to store the products in the actual greenhouse instead of an office or cooler. Boxes should be opened to prevent the buildup of carbon dioxide and be placed under a bench or trough to keep them out of direct sunlight. Microbial products can be stored until their best before date. Make sure to follow the guidelines on the package, since many of these products need refrigeration. Release methods will vary for different products. Don't shake tubes and bottles. This could damage the biocontrols inside. Instead, gently roll them to mix contents. For crawling biocontrol agents, like mites, sprinkle a small amount on the foliage. Product that falls off onto the bench or floor is wasted product, as most crawling biocontrol agents won't be able to make their way back up. Crawling bios need plant-to-plant contact to disperse. If plant canopies are touching, releases can be made by broadcasting the product at regular intervals along the row or bench. Instead of manually broadcasting, it is possible to blow mites in vermiculite or brand-based products. Some biocontrol companies sell special mite blowers, but there are growers who make their own. Whichever blower you use, you will have to calibrate it by checking for the presence of live, intact mites on the crop at different distances, since some blowers can damage or kill the mites. If plants are spaced apart, other tactics have to be used to ensure proper coverage, such as using individual mite sachets on each pot or plant. For flying biocontrol agents, less release points are needed as they can generally get where they need to go. Apply loose product within the plant canopy to ensure they don't fly straight up and out of the greenhouse. Be aware of the direction and strength of the airflow, as these insects are easily carried off or may refuse to fly if there's too much wind. You may want to release them at dawn or dusk when there is less ventilation happening. Instead of sprinkling, you can use release boxes to prevent material from falling off the plant. Put only a thin layer of material inside each release box. Distribute the biocontrols evenly throughout the crop and make extra releases directly on the plants in pest hotspots. When using parasitic wasp cards or mite sachets, hang them directly on the plants. It is better to hang them on the stems, not on leaves that may fall off or be removed. If using cards in a propagation area with mist, hang them on a stick covered with a styrofoam cup to prevent them from becoming wet. Correct placement of sachets in production areas is important to optimize performance. Place them within the canopy and out of direct sunlight. This provides optimal humidity levels in a sachet so they keep producing over several weeks. Sachets placed in the sun will peak after 1 to 2 weeks and may not give your crops the long-term protection you expect. The sachets already have a small release hole. Do not enlarge the hole or tear open the bags as they will become too dry. In potted crops, don't allow mite sachets to touch the surface of the growing media. This can lead to excess moisture, and the sachet contents may rot, which will also limit their lifespan. Another option is to use foil sachets that are more water resistant. Some biocontrols can be released using banker plant systems that provide either non-pest spray or an ideal crop for reproduction of biocontrol agents. Additionally, supplemental food, such as pollen, Ephestia eggs, or Artemia cysts, can be purchased commercially to help predators establish and reproduce. More information on the release methods or instructions for specific biocontrol agents can be found on the websites of the biocontrol supply companies.
All right, guys. Thank you for sticking through that. That was a lot of back-to-back videos.
Here's a brand new, better way to sell your house. So if you're thinking,
so I'll be including the link to the quality guide that Vineland put together, and so they basically have detailed that based on a bunch of different BCA options. So that's pretty cool if you want to get into the specifics of what you are purchasing. So we're just going to do a quick summary of why natural enemies might be failing or why we might be not be seeing the performance that we would expect. From this point forward, we're kind of making that assumption that, everything looked good after it was shipped to our farm, and that we've kind of done that quality check and that everything looks okay. So first step is we just want to make sure that we're picking the right agents for our pest problem and our situation. So you'll notice we have a bunch of predators in the red column here, and those have been broken down by types. So type 1 is more of a specialized mite predator. Type 3 is going to be more of a generalist, and type 2 is selective on a certain type of mite that typically puts out a heavy webbing. And so they've also sort of broken down that food source here as well. So obviously, if we're talking about a specialized mite predator, as we are with Persimolis, for example, on that first row, the only food that it's going to be targeting is two-spotted spider mite. Then if you look at something like Californicus compared to Phallusis, they are both selective for heavy webbing mites, and they both have the same food source. But the difference here is that it may not be as successful in all, may not be successful depending on what your production area looks like. So for example, Californicus it says that it does well in all except for fields in a cold area. So if you have low-lying areas or we're looking at shoulder seasons or it's just been sort of uncharacteristically cool for that time of year, you might not be seeing success in that Californicus. Alternatively, if you're looking at Phallacis, it's meant for field use, not necessarily greenhouse or tunnel. So I thought that was a great slide from a NARBA or NASGA presentation a few years ago. All of these agents also have sort of ideal temperature and humidity ranges, so we might have made the correct choice for our predator, depending on what that insect pressure is, but also making sure that the growing environment, not only are we talking about humidity in this case, but also temperature. So something like Persimolis will start at the bottom here, has a range of about, say, 15 degrees to about, let's say, 30, but requires a high relative humidity greater than 70 to 80%. Typically in Nova Scotia, we're looking at these higher humidity ranges, so looking more towards Cucumerus, Swirsky, and Persimolis, which are very common ones you've probably heard of before. But if you know that you do have lower humidity periods, then maybe it's worth looking at something like Halifornicus or Andersoni. And so this really is going to continue to sort of drive home that point of the importance of monitoring our temperature, monitoring our humidity, and not only from sort of a right now, immediately what's going on in here, but also having that ability to look back. So if we find that our biocontrol agents aren't working well, if we look at that sort of humidity range or we look at the temperatures that we experienced over that time, if it's low or it's not where it should be for our predator, that could be the reason why it didn't perform as well as we would like it to. So maintaining sachets, this is a bit of a review as to what was in the video. So the photo on the bottom here, we can't visibly see any holes or tears or places for those BCAs to get out. But we just have to trust that it was formulated appropriately and that everything will be able to emerge as it needs to. So making sure you're not ripping or tearing those sachets unnecessarily is very important. Especially where the sachet is sort of a slow release, supposed to last for multiple weeks. That humidity inside those sachets is important. And also making sure that they don't get excessively wet. So it's quite easy to kind of stick those stakes right into the surface of the substrate and then sort of have that sort of soak up the body of that sachet. And also just be aware of your rodent pests as much as we already are to make sure that our crop is doing okay, but they can sometimes like to chew on these sachets as well. And so they did some work with, again, OMAFRA and Vineland Research and Innovation Center on sachet placement. Basically, when you are putting that sachet in the canopy, you're actually allowing that slow release to happen. And you'll see that those peak populations will be reached around week 3, which is kind of what we hope for, compared to a more exposed sachet, which is not protected from direct sunlight and probably has less humidity in there as well. And then we also have to be aware of where we're putting them. So on the outside of the pot, like in this first photo, they don't really have great access to the actual canopy, and it's going to be really difficult for the majority of those to actually get into the canopy and work the way that we want them to. You'll also notice in this earlier crop production stage on the photo on the right, there's a decent distance between some of those sachets and the actual plant material, and then the canopy itself is actually not overlapping. And so when the canopy is small, like I said, broadcasting might be a better strategy for those early stages. And the other thing that we can look at doing is actually sort of placing a piece of paper, say over this pot on the left, distributing your BCAs, and then leaving that overnight. And then you can come back the next day, grab the paper, no harm, no foul, but it kind of keeps you from just dumping the product either on one of the 4 plants in this pot on the photo on the left, or prevents you from just dumping it on the soil. And so both of those would definitely impact the quality of your biocontrol that you just released. So that's kind of a cool strategy. If you are distributing from a direct container, if you can leave the container in the crop, including the lid, that's a great way to make sure that everything that was in there is actually getting out. They really like to hang out around the rims of bottles and the rims of lids. And so we're just really trying to maximize that transfer from that container into the crop. And so we've got a few examples of how people have left those out. If you're looking for predator-specific information, I've just included some screenshots from the BioBest app. This isn't an endorsement necessarily, but just kind of showcasing some of the information that these suppliers do have. So this I thought was pretty fantastic, and we have a few sort of zoomed in screenshots here. So we have a summary of what we've purchased. So for example, we've got Californicus, and that targets spider mites, broad mites, and cyclamen mites, and we can basically use it in all crops. We're looking at fruit, vegetables, ornamentals, medicinal cannabis in both outdoor and greenhouse environments. It talks about what it is. So we have a predatory mite, and then again, sort of outlines that it is a control for spider mite species and that it's less sensitive to warm and dry conditions. So that's always good to know, and that it can tolerate cold conditions. So it gives you some information on when it's going to perform well and when it might not. And then it also talks about the mode of action. So like we said, it's a predatory, so we're mainly eating those nasties. There's also a handful of product specifications, and that's just going to give you some information on how many sachets you're getting or the volume of those BCAs that you would be receiving, depending on what you purchased. There's also some storage information here. So it's going to talk about those ideal temperatures and sort of what you should do immediately upon receiving that shipment. On the second page here, they have some dosing rates depending on what situation we're in. So if it's ideally, we're looking at a preventative system. So we're releasing before we even have an issue, and they have that both for a broadcast or a sachet system. But if we're looking at a high curative situation, then obviously we want a much higher concentration of those individuals sort of in those hotspots and the areas around those hotspots. And so that is really going to make sure that that product is more effective as opposed to you releasing in a preventative way when really you should be releasing in a curative. Instructions for use, then they have sort of, again, you should be using it as a preventative. We have a few different release methods and the conditions that you should be releasing that in. This I think was also quite helpful, is that it outlines the different life cycles and appearances. So if you are able to get your hands on a microscope, these are kind of the things that we're looking for. We can see all life stages, and it tells us the best way to monitor for them as well. So adults are maybe found in flowers feeding from pollen, which is a good thing to know if we find that we're not able to find them. You should be looking for mobile stages underneath leaves. Eggs are laid on leaf hairs near the junction of veins. So really just try to focus your time and your energy on the places that these are probably hanging out. Finally, just before we wrap up here, I just wanted to touch on how we basically integrate our biocontrol programs with potential conventional chemistries. So it seems that the major insectaries are coming up with apps as opposed to having websites, so I can't necessarily guide you through this process, but I can kind of show you a few screenshots from what I was able to pull up on my phone. So it's great, you know, if we are thinking about controlling mites and thrips, for example, so we might be looking at this Californicus and Cumarus combination. What do we do once we have disease starting to crop up? Or what do we do if we start to see an outbreak in either these pests or other pests, and we need to come in with a conventional solution? And so what you can do is you can type in basically the systems that you have, and you can do multiple, which is great. You don't just have to do it one by one. And then you can select your active ingredient as well. And so I've included a few examples here. So copper, for example, is non-toxic for both Cumarus and Californicus. And so we see that the impact on your BCAs is going to be less than 25%, which they have given a green box. If we look over to the right, I included a miticide, so abamectin. Obviously, that's going to be moderately toxic to toxic for Californicus, and then Cucumerus we're seeing toxic, which is meaning that over 75% of your population will likely be wiped out by that. So at that point, then we just have to make a decision. What are we trying to prioritize here? Is it going to be solving the problem, or is it going to be trying to maintain that biological program that we might have invested a lot of time and energy into up until that point? The final one I'll show here. So I chose an active ingredient that is commonly used to control something like botrytis. And we can see that there is toxicity depends on the way that it has been delivered to the crop. So if you're spraying or mistblowing compared to a fumigation, we're going to see a different impact on those mites. Now, in this case, it's a bit frustrating because it looks like one method, if we spray with a Californicus system, that's not toxic, but that turns to be slightly toxic for a cucumeris, and then the same or the opposite for fumigation. So we kind of have to prioritize, our thrips our biggest issue, our mites our biggest issue, or maybe we just look for a different product altogether. All right, folks, so that's all I've got for you today. Our next meeting is going to be July 24th, and that's going to be another roundtable/office hours. And then we have a panel discussion on greenhouse structures coming in August. I appreciate you guys hanging out, and I hope you have a good day. Stay cool, guys.